Hello everyone, my name is Tanush Reddy. I am studying in Sri Venkateswara College of Engineering and I pursued in CSE Cybersecurity. Now I am going to talk about the topic on human blood substitute. Hmm. The quest for human blood substitutes. Here are some of the points. Addressing the world blood storage crisis demands innovative solutions. Human blood substitutes promise to revolutionize transfusion medicine by providing safer, more accessible options. Beyond addressing global shortages, they aim to eliminate risk associated with transfusion transmitted infections and transform emergency trauma care, especially in remote regions. This breakthrough technology is poised to redefine how healthcare systems manage blood supply, ensuring more lives are saved through reliable oxygen delivery across diverse medical settings. Now, let us go to the next topic, that is the critical need for blood substitutes. The first topic is global blood storages. 70% of countries worldwide face insufficient blood supply, severely impacting patient care and emergency response. The next concept is transfusion risk and limitations, where even with 118.5 million donations, the risk of adverse reaction ranges between 1 to 3%, compounded by the limited 42-day shelf life of red blood cells. Need for safer alternatives. De- Development of universal pathogen-free oxygen carriers is critical to mitigate these challenges and improve transfusion safety and availability. Next topic is oxygen therapeutics. Hemoglobin-based oxygen carriers. The source and modification are extracted from human or bovine red blood cells, modified to enhance safety and circulation time. Examples products like Hemophor, that is bovine origin, and Oxyglobin, that is veterinary use, illustrate clinical promise, clinical impact. Advanced encapsulation techniques limit toxicity and rapid clearance, showing encouraging results in trauma and surgical settings. Perfluorocarbon-based oxygen carriers. Oxygen distribution can dissolve large volumes of oxygen, facilitating transport through microwave culture. Chemical stability. Highly inert, minimizing reactivity and potential side effects. Challenges. Early formulations faced issues with retention and required elevated oxygen levels for effectiveness. Artificial red blood cells. Biometric design. Utilize liposomes or polymer structures to replicate the behavior of natural red blood cells. Oxygen delivery. Encapsulation of hemoglobin or synthetic oxygen carriers enhances oxygen transport with reduced toxicity and longer circulation. Research progress. Current studies focus on optimizing oxygen loading and unloading to improve clinical efficiency. Acellular versus cellular oxygen carriers. Acellular carriers, that is, modified hemoglobin or perfluorocarbons for oxygen transport. It is easy to produce, store, sterilize with long shelf life. Rapid oxygen delivery, even in constricted capillaries. Potential risks of vasoconstriction or tissue accumulation. Cellular carriers: artificial red blood cells mimicking natural red blood cells using liposomes or polymers. Better biocompatibility due to cell-like structure and controlled release. More complex and costly to manufacture, scale, and maintain stability. Potential for immune interactions if not properly masked. The oxygen carrying challenge. Hemoglobin-based oxygen carriers. Advantages includes natural oxygen binding and efficient oxygen release. Risk is vasoconstriction due to nitric oxide scavenging. Potential toxicity from free hemoglobin. Example, Hemopore uses bovine hemoglobin cross-linked to prevent rapid degradation and enhance stability in circulation. Perfluorocarbons. High oxygen solubility allows for carrying significant amounts of oxygen but require oxygen co-administration to be effective. Challenges: poor miscibility with blood, leading to emulsion, instability, and long retention times in the body, potentially causing tissue accumulation. Key matrices is oxygen affinity measured by P50 indicating the partial pressure at which hemoglobin is 50% saturated. Viscosity affects circulation and oxygen delivery efficiency. Must be optimized to avoid circulatory impairment. Retention time impacts safety with longer retention potential leading to adverse effects and shorter retention reducing efficiency. The next topic is biocompatibility and safety. Ensuring the ensuring the blood substitutes are safe and well tolerated in paramount. Here is a breakdown of key considerations for each type. Artificial red blood cells, that is designed with phosphorylated bilayers mimicking natural cell membranes to minimize immune response and prevent opsonization. Surface modification with polyethylene glycol further enhance biocompatibility and reduces protein absorption, prolonging circulation time. Hemoglobin-based carriers require stabilization to prevent rapid degradation and toxicity. Cross-linking with molecules like glutarate or diaspirin deviates reduces nitric oxide scavenging and vasoconstriction. Fluorofluorocarbon emulsions. Emu- Emulsified with biocompatible surfactants like fluoronic F68 to improve miscibility and prevent aggregation. Careful control to particle size and dose is essential to minimize tissue accumulation and potential for inflammatory reactions. Oxygen co-administration is necessary due to limited oxygen binding capacity. A key consideration for blood substitutes, that is oxygen transport efficiency, maximizing oxygen carrying capacity and releases at the tissue level. Biocompatibility, minimizing the immunogenicity and toxicity to ensure patient safety. Practically, long shelf life, stable storage, and scalable, cost-effective manufacturing process. Flow properties, appropriate viscosity for smooth circulation without intensing vascular impactions. The next topic is challenges in blood substitute development. Vasoconstriction tissues. Some HPOCs is include blood vessel constriction, potentially reducing oxygen delivery. Limited capacity. PFCs have lower oxygen carrying capacities compared to natural hemoglobin. Toxicity and immune risks. Immune reactions and toxicity remain hurdles delaying approval and widespread use. Regulatory and trial complexities. Rigorous testing and varied clinical conditions prolong development timelines. Research advantages and ongoing research. Genetical engineering and nanoparticle carriers. Allosteric modifiers. Clinical trials. Future directions and potential applications. Emergency medicine. Rapid oxygen delivery in battlefield. Remote trauma settings saves lives. Anemia treatment, offering alternative to traditional transfusions for chronic anemia patients. Organ preservation, enhancing viability during transplantation with improved oxygenation. Adjuvant cancer therapy, improving tumor oxygenation to increase radiotherapy and chemotherapy effectiveness. Wound healing, accelerating tissue repair through better oxygen supply. The future of blood transfusion, eliminating blood shortages, storages. That is, blood substitutes promise an unlimited supply, reducing reliance on traditional donations, reducing infection risks. Synthetic production eliminates the risk of blood-borne pathogen transmission. Global accessibility. Blood substitutes will improve transfusion access in remote areas and developing countries. Thank you for exploring the human blood substitutes. We appreciate your interest in advancing life-saving oxygen therapeutics. Your engagement helps drive innovation in synthetic blood development, discontinued innovation, global impact, next steps. Those are collaborating fuels, breakthrough in blood substitute technology, improving access to safe transfusions worldwide remains our goal. Stay informed about clinical trials and emerging therapeutics. Thank you.